My right hand is controlled by the left side of my brain, and my left hand by the right side.
But what does this even mean? And are there other things linked to one side or the other too? Using experiments you can do at home, I'm going to show you how strange your asymmetric brain actually is.
And then I'll ask why it's split at all.
And once you understand why your brain has evolved like this, you'll never think about it in the same way again.
Because having a split asymmetric brain isn't a flaw. It's an evolutionary masterstroke. Here's a demo you can do at home to really feel this left-right brain thing. Take a piece of paper and then a pencil in each hand. Draw a square with one hand and a circle with the other at the same time. Pause this film and do it now.
And go.
Interesting. Very
Oh, really nice circles.
That's a square.
So, how did you do? It's really difficult, isn't it? Well, it turns out that this difficulty is completely normal.
Your brain has two hemispheres. Both are trying to run their own motor program simultaneously, one for the left and one for the right. And that's challenging.
Because the pathways linking the hemispheres can't coordinate each other's instructions without information bleeding across into each other.
This is called bimanual interference, and it's clearly showing the effects of having two brain systems.
Incidentally, if you had no problem with this task, then lucky you. That's not bad or weird, but you are in a small minority.
I definitely think
For the great majority of us, the task is surprisingly hard.
Go in the same direction.
And you probably also found that one hand was better than the other.
Pretty good.
For me, it was definitely the right. Obviously, what we're talking about here is handedness. Now, most of us are right-handed, so there's a natural manual dominance on the left-hand side of the brain.
So what's all of this actually about? Why do we have left or right brain dominances? Is each hemisphere different, or is it all programming? Let's start with the brain.
Here's mine. It's actually a life-size copy of my brain I had made from an MRI scan for a recent TV series.
You can see it's made up of all sorts of shapes and bends and swellings. It's also very clear that it's a shape with two halves, a mirror image of each other, almost. So why is that?
The answer to this arguably came in the 1960s as a result of some groundbreaking work from experimental neuroscientist Roger Sperry and his student Mike Gazzaniga.
They already knew that left field of vision was processed by the right brain hemisphere and right vision by the left. But how did that work in practice? And how did vision relate to language? We spoke to Gazzaniga recently and asked him what they were trying to understand.
If you look at the brain, there seem to be two halves. And some people just ask questions, well, wait a minute, how does information from one side get to the other? How is this whole thing what's responsible for our behavior? How does this work?
At Caltech in California, Gazzaniga had an opportunity to explore this with the help of some unusual patients. Patients who'd had what we would now consider barbaric surgery to help them with epilepsy. Surgeons had literally severed all the connections between the left and right brains.
So in general, and in many kinds of epilepsy, there's a focus. And for some reason, there's an underlying pathology that creates seizure activity in the brain. And then once it gets triggered, it gets transmitted usually across to both hemispheres of the brain. So the thinking was real simple. Let's keep it isolated. Let the seizure occur, but only the hemisphere that it is in would seize up and cause the convulsion. And the other one would be seizure-free and stabilize the body. And therefore, there would not be a general loss of consciousness and all the rest. That was the simple idea behind it.
And because of this, Gazzaniga saw a unique opportunity to explore how the two brain hemispheres differed. The first patient he worked on was nicknamed MJ. He had such appalling seizures, he was ready to try anything.
Gazzaniga's first experiment with MJ was pretty simple. He would have him focus on a central dot. Then he would flash up shapes on the far right and far left. Now, we are forward-looking creatures, so both our eyes have a common field of vision. But if you have a shape on the far left, then it won't be visible in your right eye's field of vision.
First of all, we had a chance to test him prior to his surgery. And he behaved just like you and I would behave. If you showed something to one part of his visual field, the other side knew about it. Just put a thing in one hand, put an object in one hand, the other hand knew what it was. Just normal stuff because his brain was normally interconnected.
Then MJ had the operation. Now, it sounds absolutely barbaric, but actually, it almost completely cured his epilepsy. This part here, the corpus callosum, which links the two halves of the brain, was completely severed. Almost everything we know about the left and right hand brains came from what happened next.
So he comes out to Caltech, and I have my room all set up, and I said, okay, Bill, is his name, fixate on this. I want you to fixate on this little dot on this screen. Just stare at it. And he said, okay. And the first thing I do is flash a picture of an object in his right visual field, which goes to his left dominant speaking hemisphere. And he says, I saw a square or a box or a circle or whatever it was. If you name it, everything looked just like you and me. So then you would say, okay, I'll fixate the dot, and then you would flash something to the left visual field, just on the other side of where you were looking, right? And that went to the right half brain, right? Anatomy. And he said, I didn't say anything. And I can tell you, you kind of break into a cold sweat when this happens. And so I said, okay, I want you to point to where you see something, right? And use either hand. I don't care which hand you. And again, the right visual field, left hemisphere, right hand worked perfectly, just like you and I would do it. But then suddenly also, when I just flashed the dot to the left visual field, right hemisphere, left hand, it pointed exactly where the dot was. So that tells you that the visual motor system was working just fine in the right hemisphere. But it was no longer connected to the speech center, and the speech center couldn't tell you about it.
Just think about what's happening here for a moment. In a normal brain, when we see an object on either side of our visual field, we can both point at it and name it. But here, because his hemispheres were separated, MJ's right hemisphere's motor function was fine, so he could point at an object with his left hand, but he couldn't name what he was looking at. Speech seemed to be locked in to his left-hand brain.
Next, he had MJ try to solve a simple puzzle, just using one hand.
All you're supposed to do is arrange these four black blocks so they match that pattern. And we're going to first ask him to do it with his left hand. And you'll recall his left hand gets its major control from his right hemisphere. Okay? So we're seeing basically the right hemisphere at work here. And there he is. He's got it. So that's his right brain working. Now we're asking him to do the same thing with his right hand. Same thing. And now his right hand is getting its control from the left hemisphere, the one that speaks to us, the one that knows language. And it just doesn't have this skill. It simply doesn't. The right hemisphere controlling the left hand is good, but the left one that's doing all the thinking is terrible. And that right hand just can't do it. Look at, he even arranges them in terms of 3 blocks and 1 block. He doesn't even square them up to the 2x2 nature of the question. And now watch, his left hand's watching the same thing, and you begin to see him move. I'm going to come help, he says. There it goes. And take it back and say, no, you can't do it. Just try to solve it with your right. And all of what that might signify was born with this simple test.
This was the first clear evidence that the 2 hemispheres are like 2 separate brains, each with different skills and strengths. It means that our earlier drawing experiment was a bit like 2 brains fighting for control.
This discovery shot Gazzaniga to scientific stardom, and the research became the cornerstone of modern neuroscience.
We're talking about 50 years ago, and we're talking about a moment where I knew completely well what it meant in the context of the time. But, as I said, that's 50 years ago. I still think about it every day.
To explore the most recent research, I've come to the University of Sussex to meet neuroscientist Professor Gilly Forrester. She's obsessed with the work of Gazzaniga.
Oh, excellent. So we're looking at some footage that's at least 50 years old by Michael Gazzaniga. And
it's almost like 2 different people. The right hand is...
yeah, but you see, they're fighting over it, aren't
left
they?
hand said, oh, hold my beer.
But yes, but it's fighting to get on the table there.
Yes, quite sort of independent.
Because it knows how to solve the problem. It's likely this is a spatial task that will require more right hemisphere, and that left hand really wants to get involved.
Okay.
Okay, so this was the beginning of really us gaining an understanding that the 2 sides of the brain have these nuanced dominances, and it really set the field in motion.
Gilly's put together a series of fascinating experiments which will reveal our modern understanding of brain asymmetry. And they're so good, they're definitely worth trying at home to understand your own brain. Gilly's first experiment was very straightforward, placing pegs into a grid.
I'm going to start the clock in just a minute. You place both hands on the table. Again, as many as you can, as quickly as you can. Are you ready? You steady? Go.
If you want to try this at home, get some pegs or a jar of coins, or in this case, a jar of Scrabble pieces. Then, against the clock, arrange the coins in size, or here, arrange the pieces in alphabetical order. In fact, it would work with anything involving a collection of items that you pick out and arrange by hand.
And 5, 4, 3, 2, 1, and stop. Super. I'm going to stop the timer, stop the video. You've done quite well there. But what I was actually looking for was to see how many times you used your right hand versus your left hand. So I'm interested in working out if you have a bias for which hand you prefer to use for these kinds of tasks. Now, I don't even have to look at my video to know that you were absolutely right-handed on this task.
I think I left my left hand
I
on
think
the
you
table.
did. I think you did. And that's not uncommon. And this is a reflection of your preference for these kinds of fine motor actions.
So how did you do? Clearly it shows if you're left or right-handed, but hopefully it'll reveal that handedness is way more subtle. Because some people, like me, use just the right hand. Some will use just the left, but many will use both hands.
Could you explain to me why you think you didn't use your left hand?
Well, I was going for precision, so I'm right-handed, so I assumed with my right hand I'd be more careful in putting the pegs in.
And indeed, this is demonstrating that bias in your right hand that's tied to your left hemisphere dominance for this type of fine motor behavior.
So other than confirming that I'm right-handed, what does this tell us about my brain?
So all of us humans, we have this gorgeous pink squishy brain, 2 hemispheres, and they look rather symmetrical. But actually each side has its own slight bias for specific kinds of behaviors. And fine motor coordination seems to be dominant for most of the population in the left hemisphere, and it manifests in the right hand. But handedness, even though we know it to be part of our identities, left-handed, right-handed, maybe we're ambidextrous and can use both with equal precision, it's more complex than that as well, because your handedness can also be mild, moderate, or strong, going from left to right. And this tells us a little bit about your individual brain and the way it is organized and the way that it functions.
So I'm obviously right-handed, and about 90% of the population are too. 10% of us are left-handed, and then there are a few who switch hands depending on the task. There's clearly some sort of asymmetry, but it's far from binary, because this asymmetry runs much deeper than handedness and fine motor control.
The next experiment will reveal another deep asymmetry, and it tells us how we see and maybe even how we think.
We're going to look at some faces. All I'd like you to do is tell me which face seems more expressive to you. You'll just have 2 choices in each trial.
These are the pictures. Which of these 2 is more expressive? What do you think?
Yep. Well, let's begin.
A.
Thank you. And here.
B.
Excellent.
I think A.
So the really interesting thing about this test, it's called a chimeric face test. You might see these images and think, oh, that looks a bit weird, right? It's kind of one side of the face is neutral, the other side of the face has an expression. And each trial, I show you 2 stimuli, one where the expression is on the left and one where the expression is on the right. But actually, each of those stimuli are absolutely identical. The only difference is that it's mirror imaged.
Right.
However, your brain will see one of those as more expressive than the other based on which visual field it's falling into. So for the majority of the population, if we see the same stimulus on the left side and it's a face and it's smiling, for example, it will look happier when it's on the left side than when we see it on the right side.
Right. Okay.
So, how did you do? If you got mainly Bs, then you're in the majority. Expressions on the left side look stronger than the right. Years of research indicate that for most people, the right hemisphere is more specialized for emotion and facial processing.
Jim, you showed the classic right hemisphere, left visual field bias for processing faces.
Right.
So when you had a choice, you more often than not, and you show a left visual field dominance for recognizing faces and their expressions, and that suggests a right hemisphere dominance for this capability. And
that's normal.
That's very
weird.
much.
Okay,
That's reassuring. Like most people, my right brain is better than my left at reading and expressing emotion. But there's more to come.
In the next experiment, I'll show how the way we process faces reveals another asymmetry. Faces dominate our emotional social interactions, including the way we nurture our young. Experiment 3.
For this next task, I'm going to hand you different items. And each time, I just want you to take the item from me and hold it in your arms like this.
Okay.
Okay?
You might not be able to do all of this next test at home because some of the props are a bit involved, but you can get a sense of it with a doll and a cushion. Also, try it on someone else as well. Don't use a real baby, though.
Right. It's a rather heavy item.
Okay.
I just want you to take it and hold it where it's most comfortable for you.
Okay.
Perfect. Does that feel okay?
Yep.
Brilliant. Okay. So I'm going to ask for that back, and I'm going to give you the next item. You ready?
Yep.
Here you go.
Okay.
Thank you. Does that feel okay?
Yep.
Fantastic. I'm going to take that one back too. One more to go. Fantastic. We're done.
Great.
Is that okay?
Yep.
All right. I'll take that one back from you. We can have a chat.
It's been a while since I've held a baby, so...
So, just out of curiosity, when I handed this one to you, do you remember how you held it?
This was the first one?
This was the first one.
Yeah, I think, because you said it's heavy,
I did say it's heavy.
and because I'm right-handed and I favour my right arm, I think I cradled it under my right arm.
And you did. And that would be in line with what the majority of the population would do as well. This is an inanimate object. It's not that different from holding on to a sack of potatoes or a big sack of flour. Right. It actually weighs about 5.5 pounds. It's full of fishing weights. Right. It is no different at all to this one. So this is the one I gave you second.
Right.
Same fabric, same weight. Only difference.
Got a little
this
face on it. Or,
isn't it a face, though?
I mean, I saw it as a face. It's an oval with 3 black dots.
And remind me how you held this one.
I can't remember. I think I may have switched.
You did switch.
I may have switched. I think I may have held it like this.
You did switch, and you immediately held it like a baby. Like I said, exactly the same fabric, same dimensions, same weight. Yep. This is what we call a protoface.
Right.
3 dots and a boundary around it. This is enough to elicit a side bias in holding that is different from the kind of holding you would do of an inanimate object. The inanimate object, you went to your stronger arm, like you said, you're right-handed, you put it in your right arm, and you would probably carry it around like this, or maybe over your shoulder like this.
Yep.
As soon as you see this protoface, you've treated this like an animate object. This is now a baby. You've placed it in your left arm.
See, I didn't think, I mean, I sort of recognized that subconsciously as looking like a face. I'd assumed I switched arms simply because I knew, maybe because you didn't say it was so heavy. Maybe I thought for a bit of variety, I'll switch it. But maybe subconsciously, you'd just given me a baby.
But what's even more interesting is that you don't have to have had any experience of faces or babies to express this left side cradling bias like you've done. So if I bring our baby back out again, you immediately held that baby with confidence in the left arm.
Yeah, it's been a few years. My kids are grown up now. But
and if I asked you, why did you put this baby to your left side? You're right-handed, baby's pretty heavy. Why did it go to the left?
I think it's because you need the dexterity of the right hand to
To do things.
To do things, to
Because
feed it, to
right-handed.
wipe its nose, to pat it. The left hand is just there doing nothing, holding the weight.
So interestingly, we've been doing a lot of studying around this area. This has been quite a hot topic of study for at least 2 decades now. And left-handers also prefer to hold babies on their left. But if you ask them why they do it, they say because it's their stronger arm. What we now know is that the left holding bias is not human unique again. It's playing off of that left visual field bias for processing faces and emotions.
Nothing to do with the weight of the
Nothing
baby.
to do with the weight,
or the
baby.
dexterity of your dominant hand to do stuff.
They may be the influential factors, but they're not the driving force. So your ability to look at this underdeveloped, vulnerable baby and check to make sure it's okay, you are faster and more accurate in telling whether or not this baby is okay with your left visual field.
Right.
So putting baby there. And remember how I showed you that it's not just about perceiving, but it's also about how we produce our expressions?
Yeah.
Well, you're more likely to be producing a stronger expression on the left side of your face to present this to the
baby
infant.
to see.
Exactly.
Well, I'm presenting it with my more expressive side.
You are, and babies are born with very underdeveloped motor sensory skills, so their ability to see far away or see strong signals is limited. So by getting your stronger expression into their eye line, you're having this mutually beneficial relationship here.
This is a hugely profound finding, and it's borne out by years of research. To recap, most people, including left-handers, will cradle a baby in their left arm, which tells us it's not about strength and it's not about dexterity. It's a perception bias. It's about which side of the brain best delivers and receives emotion.
50 years ago, Gazzaniga discovered how language was primarily left-sided. Now we've discovered asymmetries in manual dexterity, emotions, reasoning, and even behaviors like nurturing. But the latest research is now revealing that it's not just humans that have this asymmetry. It's everywhere.
And for a really long time, we thought this was just about humans. We're unique, we're sophisticated, we have these behaviors that no other animals have. But in the last kind of 30, 40 years, we've been doing a lot of looking around other animal species to see, do they also have these different sorts of behaviors that are biased to one side of the body?
So whether their brains are asymmetric like ours.
This is Hulk. She's a cane toad. Our ancestors split from her ancestors some 350 million years ago. So the things that we have in common with her tell us a lot about what life must have been like back then. So here's the million-dollar question. Do toads have brain asymmetry?
Now we're going to place one of these worms, and we're going to put it on the wheel. And what we're going to do is rotate the wheel around, and we want to see at what point Hulk is going to go for this.
So when it comes into vision.
Exactly,
Right.
exactly. So shall we pop it on the tray?
Okay.
So yeah, you can see all these.
Let me take
You want to
one.
go for it?
So I'll put one on this side then.
Yep, and we're going to rotate it round, and
Oh.
we're going to see at what point she wants to go for it.
There we go.
Oh.
Oh,
So many toads over many trials have shown us that they actually prefer to wait until the prey comes into the right visual field. So this right eye, which connects directly to the left hemisphere.
so the right eye is better at detecting food as it comes in.
That's right, exactly. So the right eye is where she will be faster and more accurate at getting that worm. This is suggesting to us that Hulk has a right eye preference, left hemisphere preference for these feeding behaviors.
And this has been observed across many kinds of animals too, from fish to birds and even some insects. And now research is suggesting that even primitive organisms exhibit some kind of asymmetry too. This is C. elegans, a tiny worm, just a millimetre or so long. Worms a bit like this evolved over 600 million years ago. Recent research has found that C. elegans has a receptor specifically sensitive to sodium on one side of its head, while the receptor on the other side is sensitive to chlorine and potassium, suggesting that some sort of neuronal asymmetry is almost as old as life itself.
What is the reason for this asymmetry to have emerged in the first place?
Yeah. Well, from all the animal work, we do think that this is all about the animals being able to eat and not be eaten at the same time. So if you think about an individual who, like our cane toad today, who has the eyes on the sides of her head, if she's able to use one eye in the opposite hemisphere to take charge of looking out for food, while the other eye and hemisphere takes charge of looking out for predators, then she can do both things at the same time. And these are the most important activities for an individual to stay alive in the wild. So this is where we think it all started.
Having an asymmetric brain clearly has its advantages for many animals. It can allow them to parallel process, to do two things at once, like hunt for food and keep a lookout. But what is truly incredible is how these asymmetries have endured. From 600 million years ago, they're still with us today. But as time has passed, they've changed their function.
So I can see why it started then, and I can see why it's still useful for many animals today. But why has it survived in humans? How does it manifest itself with me today? Why do I have that bias?
That's a really great question, and it forms the basis for a lot of my own research. What we know is that it has stayed within us. So we do express these biases still, just like we saw with you today. And we think that these original fight or flight or motor sequencing actually form the basis of our modern human behaviors. So for example, your ability to solve the pegs actually is an adaptation from the very early feeding behaviors. That left side of the brain that for our toad was allowing her to do that feeding behavior with the worms is allowing you to move those pegs around to solve problems. So we have kept those old capabilities, and over evolutionary time, we've extended upon them to behaviors that look super sophisticated, but the actual computational power that's going on in our brains is still relying on those very old biases. And so our uniqueness actually comes from our shared capabilities with other animal species that dates back well before the rise of humans.
So what do we make from all this? Well, the key realization for me is just how old our brains are. So much about how we think is based on what happened in the evolution of basic neurons over 500 million years ago.
And the strangest part is that these ancient biases didn't disappear as our brains evolved. Instead, evolution built everything on top of them: language, reasoning, social intelligence. So our glorious, complex human brain is balanced on foundations predating the dinosaurs, laid down by creatures with almost no brain at all. The parts of the brain that once kept our ancestors alive, spotting predators, finding food, are now the engines of language, creativity, logic, and emotion.
The left and right hemispheres of our brains are relics of a deep past repurposed into something extraordinary. Because the genesis of intelligence, the foundations on which it's built, is asymmetry. And that asymmetry is still fundamental to what goes on in here today.